So today we have eye movements. Now, so we're talking about this in a sensory course because we move our eyes in order to see better. They are what keeps the image stable on the fovea and, in fact, the entire retina. And over the last couple of years, cameras have been coming, or your iPhone's cameras have been coming up with a stabilizing system. So whenever the camera moves, that is, if you're holding it and your hand is shaking, so whenever the camera rotates a bit, there's gyroscopes inside the camera that sense that rotation, and it moves the image on the camera itself, and so the image remains stable on the back of the camera. Now, we spoke before that the image is clearest on the fovea, and so some of the eye movements are trying to do that for you. So just for you to see how important that is, I'd like all of you to hit the table when you see a note. There'll be O's and C's appearing all over the screen. Don't move your eyes when they do. Keep your eye on this X here, and hit the table when you see an O. As hard as you can. Okay, there were a few clicks towards the very end. So the first ones were way off in the periphery. We can try it again if you want. Oops. None. Oh, they're appearing. Better. Okay. So, and you can see how close the O's and the C's have to get to the fovea before you start being able to distinguish. So the fovea is actually a small part of your visual field. So the next thing it does is if the image moves, or your head moves, it stabilizes the image. So again, you notice how hard it is to read this, right? While it's moving. When it's stopped, it's pretty clear. But again, if it starts moving, the image is unreadable. So we've developed 5 types of movements that attempt to address these problems. And we'll next cover what unique role each of these types of movements serve and how they manage to solve the problem. So the first one is saccades. We spoke of saccades many times before in this course. And for this, if it's possible, try to move next to someone else in the class. Form pairs. If you don't mind. Okay. Well, you have to move your laptop. No. Maybe Alex, you could. If somebody doesn't. If you're sitting beside 3 people, that's okay too, because you can look back and forth beside each one. Okay, so I want to for you to practice these movements one by one. We'll go through all the different types of eye movements. So you can see that saccades here, as shown here, they move your eye from target to target. And the vision is poor during saccades. And to compensate that, the saccades move very, very quickly. They go something like 1,000 degrees per second. So in a second, they'll wind, if they could move, rotate right around their sockets. They'd go around their sockets 3 times. That's pretty fast. Okay, so now pair up and so one of you will put your fingers like this in front of the other person's eyes and have him make saccades back and forth. Try a few fast ones. Look at their eyes. And so if you notice that if they're making, you can't see the eye move during the saccade. It goes so fast that it does move. Now try making slow saccades. If you haven't tried that, try to move slowly. So you should see that you can't make slow saccades. You should see that either the saccade breaks down into tiny little steps, but they're all fast, or that's one big fast movement. So it's kind of neat because you can make an arm movement as any other movement in your body at any speed you want. But that's the one movement that you can only make quickly. And we'll find out a little bit about why that happens. Okay, the next movement to try is something called a vergence movement. So you can see here, the target moves nearer, then farther. What happens to the eyes? Okay, try that with your neighbor. Look at one finger and then the other finger. Try to put the 2 fingers right on top of each other from the point of view. And this finger, the close finger is close as possible to the nose. So the close finger has to be really, really close. And look at the eyes. It feels funny sometimes. Because you're not used to making it. So, can someone tell me what differences they notice between that vergence movement and a saccade? One difference between the two. Yes. That's right. Yeah. So in saccades, the direction, both eyes move the same distance and the same direction. Now the eyes are moving in opposite directions. Anything else you will notice that might be different? Well, what's neat about these movements? You can actually see the move. Whereas in saccades, it went from one position to the other position. So they're a lot slower. Okay. Now we'll do pursuit eye movements. Okay. So for that, what you do is have some person track a finger back and forth in front of their eyes and look at their eyes as they're moving it. Okay, try that. Don't move slowly, 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 slowly. You don't want to make it too difficult. Yeah, don't have to move it back and forth. You can move it in a circle, up and down. Don't move it slowly. Move it slowly. Okay. So, now try making the same movement but without the moving finger. Okay. What did you see when you tried moving it slowly, or moving it out without a moving finger, I should say? Hmm? Can someone tell me what happened without a moving finger? Yes, it was a bunch of cicads instead. That's correct. Yeah, it's without practicing, you can't make a pursuit movement without something to follow, like a, a ball across the sky or a plane. You need a target. Okay, next one. So in this case, the head moves. Okay, what you'll do is rotate your head instead. Put the finger stationary and rotate your head back and forth. And okay, so notice it looks kind of the same. Okay, so to prove to yourself that it isn't the same part of the brain that's driving both, you can either take a paper, would be best, um, if you've got a piece of paper, or a finger, um, and look at the finger while you do this again. Rotate your head back and forth, and then faster and faster and faster and faster. And see the details of your finger, that they're still sharp. Okay? Now do the same thing by keeping the head stationary and shaking your finger back and forth in front of your head. Notice that you can't see almost anything. Okay? So you can't track it. So the VOR and the pursuit movements are different circuits. And then we'll see what those circuits are. And finally, if we a movement called the OKR, the optokinetic reflex. And this is activated when a large part of the world, sort of everything that you see in front of you, seems to shift. And from that you get a sense of self-motion. And we saw in the previous lecture that its role is to assist the VOR. And in some cases it can make you fool you in thinking that in fact you are moving. Okay. Now let's look at what moves the eyes. Well, we've got these muscles. They're attached to the sides, the top, and are around the eyes. And one thing that's important is that the neurons of these muscles are tonically active. And when you want to turn the eye, you contract one muscle and the opposite muscle has to relax. So then the eye turns. And you have to maintain that muscle has to maintain that contraction to keep the eye in that position. Otherwise, it'll just slide back to its central position. The other thing to note is that we have sort of 6 pairs, or 3 pairs. If you count both eyes, you have 6, but one eye only has 3. And you have 3 pairs in order to move the eyes in any possible direction. So you have muscles that pull you horizontally, vertically, or torsionally. And we can see this by passing your mouse over any of these. You can see that this muscle, the medial rectus, turns it nasally, that is, towards the nose. This muscle here turns it temporally, that is, away from the nose. This muscle here turns it upwards. Now, if you look carefully, you can see this line here. This line here is vertical and it's straight up. Now, when you move this way, it's tilted a little bit towards the nose, okay? And that's called intorsion. The other muscle pulls it down, and the vertical line is tilted a little away from the nose, or clockwise. The previous one was counterclockwise. And then we have the two remaining muscles which wrap around the eye, and they, as you expect, would turn the eye in a torsional direction. And that's because your head can rotate. You can tilt your head to one ear or the other ear, and your eye has to turn in the opposite direction. So this muscle moves it towards the ear, towards the nose, this muscle away from those. But also notice they have a little bit of vertical movement. This muscle moves it down, and this muscle moves it up. So these are the 6 muscles of the eye. They're driven by a circuit like this. We won't go into how all the muscles get activated. We'll just look at the horizontal ones. So the horizontal ones are driven by the horizontal canals. And then we saw this last time with the vestibular nucleus, and then the motor neurons, and then the muscles. So this is called the direct pathway of the VOR. We saw that last week. And we can see here, as the head starts rotating, the eyes eye muscles get contracted, and they turn the eye in the opposite direction. But notice that after the head is head turn is completed, there's no longer any extra activity coming from the vestibular nerve, okay? And so there's no extra activity contracting these muscles, and they slide back to their resting forward position, okay? So you don't want that. When you turn your head, if you're looking at somebody and you turn your head in one direction, you want your eyes to remain on the thing that you are looking at. So you need something else to help this circuit along. And that other thing is called the indirect pathway. And you can see it here. You see from the vestibular nucleus, you go to this indirect pathway. And the indirect pathway goes through this horribly named prepositus hypoglossae, PBH for short. And that nucleus, has a neuron that sends a signal to the muscles, but also back to itself. So that means any time an action potential comes into it, it reactivates it. It keeps going. And if another action potential comes, it adds to it. And a third action potential comes, it'll add to that. And gradually the activity will build up depending on how fast you're moving. So this circuit is important because it forms sort of like a short-term memory. And it keeps your eye, in this case, turned after the term is complete. So there's no longer any more signal coming to the vestibular nucleus, but these neurons keep firing and keep the eye turned to the new position. In a sense, this is doing a type of calculus. How many of you took calculus? Oh, good, a good number. So you will remember that the integral of velocity is position. Okay? So if you integrate velocity, which is what the vestibular nerve nucleus is telling you, you get position. You get the position to which the eye should have moved to. and then that position is being held by this tonic activity and held, and so you could look at the person you're looking at before the turn began. Kind of neat thing that your brain is performing calculus anytime you turn the head. So you have then these two pathways, one direct, which moves the eye during the turn, and the other one indirect, which holds the eye in its final position after the turn is complete. Okay, next let's move on to the saccades. Now we saw that only the fovea can see clearly and in detail. So when you look at a sentence, read the words in the sentence, this is what you see. You see the little dot shows you where the fovea is. And around it, everything's clear. But as you go further away, things become blurred and you can't make out the words of the sentence as clearly. What's also interesting is that every time it moves, everything becomes blurred. Even, so you lose track of the whole thing. And so, because during the movement, vision is impaired, you want the movement to be as short as possible. So you're not blind during the movement. That's not a good idea to be, not being able to see. And so, again, you rotate these eyes at these very fast velocities, and to do that, you fire these neurons, or you send a signal to these muscles to contract as fast as possible. So the neurons that come to these muscles have action potentials of the rate of 1,000 action potentials per second. So how many times do you think, on average, you make a saccade? Is it a day? 1,000? 10,000? 100,000? I think 100,000 is a little under, but pretty close. So it's amazing. I don't think you make that many steps a day. So you largely do it unconsciously, because you're doing things like reading and looking around the environment in which you are. And so you do that every time you make these saccades. You activate this thing called the PPRF, the paramedian pontine reticular formation. And again, this is near this nucleus, which is the nucleus for this nerve. And it sends out this burst to these two muscles, the lateral rectus on this side and the medial rectus on this side. And these two muscles then turn both the eyes to the left. This PPRF is also on the left. Now, you can imagine a dividing line going right down the middle. There's a mirror image circuit to drive the eyes to the right. So there's a PPRF on the right side that will activate this medial rectus and this lateral rectus. Okay. Again, you have the same problem as you had with the VOR. You need two circuits. Okay? You need a direct pathway and an indirect pathway. So when the burst comes, it turns the eye, but after the burst is complete, you want to keep the eye at that position, not have it slide back. If you move to the next word, you want to keep looking at that word. So this is the circuit that does it. Okay, then just test you on whether or not you got this. We'll have a quiz. Okay, let's suppose you had a lesion, and the lesion blocked the axon that came from this integrator or calculus machine to the neuron. So this thing removed the the tonic activity. Would it produce this movement? So this is a normal one, that's the saccade. Would it instead make a tiny little saccades instead? This movement? This movement? Yes. The brave ones raised their hands correctly. Okay, so yeah, if you have a lesion of this part here, well, the signal from the PPRF is still getting to the muscle, so it'd move it quickly. You'd have the same movement as before, the same movement, but then there'd be nothing holding it, and you'd slide back to where you started from. Okay, let's suppose you had a small lesion of this nucleus, so you didn't lose all the signal to the muscles, but you lost, let's say, a quarter of it. Okay, which movement then would you have? Would you have this one? This one? A few? This one? Okay, we'll try that again. This one. Come on, you gotta commit yourself to one of these. It's a choice of three. It's probably not this one because we already covered it. So it's either this one or this one. Which one do you think it is? This one? Ah, a few more hands. The few more are right, the brave souls. Okay, so it's a partial lesion. So you're getting not the normal saccade, but a smaller one. You're still getting a tonic movement because you're just getting a smaller tonic movement. Okay, so you make a small saccade, hold it there. Oh, I'm not there, so I might as well make another movement. Whoop, I'm not there. And you keep going until you finally reach it. Okay, so let's see what the last result is. And so you're lesioning the signal from the burst to your motor neurons. Are you producing this movement? This movement? Okay, good. You guessed it. I mean, it wasn't this and wasn't this sort of matter. Pretty obvious choice. Yeah, now you don't have this thing. So you don't make the quick movement that you saw before in the normal saccades. But you still get to where you're going because you still have this circuit, which sort of slowly gets you to where you want to go. So this pathway gets you there quickly, this pathway gets you there slowly, and the combination of the two gets you there quickly and holds it there. Okay, now we saw before that question is, what initiates saccades? So we saw before that during fixation, there was something that was keeping your eye from moving. Okay, and we saw that in the middle, the foveal part of the superior colliculus and the foveal part of the frontal eye fields, there was a tonic activity coming out during fixation. That tonic activity goes to these things called omnipause neurons because they're on most of the time and then they're briefly pause. And they pause only during saccades. So they're inhibiting this PPRF from firing, and then they stop for some reason, and the saccade proceeds. Okay? So they are, whenever there's a hill here, this thing is being activated, which in turn inhibits the production of saccades. So as long as you keep a hill there, you have to maintain fixation. What then allows saccades to produce? Well, when something, in the case of superior colliculus, something flashes, something compelling flashes in your periphery, a hill of activity, starts building up here, and at the same time gets removed from the foveal region. And when that happens, there's no inhibition here, there's excitation here, and the burst occurs and drives the eye to its new position. Okay. Now the question is, what stops the saccade? So you got the saccade going, the PPRF is firing as madly as it possibly can, but how do you stop it? Okay, well, it could be you got sensors in your muscles that tell you. These muscles are like any other muscle. They got spindles, and they tell you, when you've reached the right target. Okay, well, it's not these, because people thought that this was occurring, but they found that when they removed the afferents that come from these spindles, the saccades remained as accurate as before. And in hindsight, that is not unexpected, because these movements are so fast that even if they had a reflex, they wouldn't get there in time to stop the movement. Now, it's not coming from the eye either, because visual feedback would be even slower. Okay, it takes you about 200 milliseconds or something like that to react to anything, a visual stimulus. By that time, the eyeball were it able to do so, would have turned halfway around its socket, or more than halfway around its socket. It's going so fast. So it's not that either. Well, if you click this in your search for things to do, you find that's correct. Why is that correct? Well, this is quality discharge. This tells you what the eye's position should be. Okay, so it's an internal estimate of the eye's position. And so if you know that, you can use that to stop the eye movement. Now, you might think that it's a surprising place to put quality discharge. This is quality discharge for eye position. But it's a good place, because this is where every movement would have to go through this PPH. So we saw the VOR went through it. Pursuit movements go through it. All the movements put a signal into this PPH. Now, how does it work then with this PPH? Well, what happens is that a signal appears here, and then that sends a signal to the PPRF that produces a burst. The burst gets integrated to position. That position then moves the hill, and when the hill reaches the omnipause, the center here, the fovea, in other words, a good estimate of that the eye should have reached the fovea, it stops the PPRF, activates omnipause neurons, that inhibits the PPRF, and the saccade stops. Okay, so this internal estimate of eye position is guiding this hill across the superior colliculus. So it's like your sort of ballistic guide missile. So in a ballistic guided missile, it has an internal sense of its own position, and it steers the missile towards the target. Okay, so where do the saccadic commands to the brainstem come from? We've covered that before. We saw that there are two pathways, one through the superior colliculus, the PPRF, and then a longer one to the visual cortex, and from there to the frontal eye fields, which again go to the superior collicus and again go through the PPRF. So we have two pathways, one short latency pathway through the superior collicus and a longer latency pathway through the visual cortex and frontal eye fields. Okay, so we said that saccades are interesting because both eyes move together in the same direction, and with luck, both eyes should reach the same target. But what happens when they're not equal? Let's say for some reason this muscle here became weakened, or the signal to it got disrupted, and so it moved, let's say, half the distance it should. Okay, if that happened, you'd become strabismic. You would see double. When your eye landed on a word, you would see two different words. So if your eye you're looking around the room and your eye landed on a face, you'd see two faces. Well, what's often done in this case is the clinician comes, and to one's surprise, he or she will patch the good eye. Put on glasses or a patch, and the patch will impair the vision to the good eye. And with time, if this lesion isn't, overwhelming, the brain will start sending out bigger signals to this eye muscle, and the eye will start moving the correct distance. So these movements become larger with time. But at the same time, the eye behind the patch will start making bigger movements that are twice as big as they should be. Surprisingly, when the patch is taken off, the good eye gradually reduces the size of its movements. And the signal for that recalibration again comes from our old friend, the cerebellum, in this case, the vermis of the cerebellum. How it happens to do this, that it only affects this one muscle, only affects the muscle that has been damaged, we don't know yet. But it will happen unless one of two things occur. One is that it exceeds the capabilities of the cerebellum. That is, if you've got a muscle that's too weak and no amount of increase in its firing rate will help you, or the cerebellum itself is somehow impaired. You've got sort of damage the cerebellum. And one thing that seems to impair the cerebellum is having too much to drink, and that's why after drinking too much, you start losing your balance and stuff. Okay, now let's look at nystagmus. Nystagmus is this back and forth movement of the eyes. So one direction is slow, the other direction is fast. It happens in all kinds of things. You can see here there's a turkey walking down my backyard, and you can see it's got this movement here and a fast movement. It's got a hold and a move. If you look during the hold, its head is stable in space. Okay, its keeping its body is moving forward, and its head is just stationary. And then it flicks its head forward again, and then its body catches up, and then flicks its head forward again, and its body catches up. So it's like acting like the VOR, keeping its eye stable in space. Well, your VOR does the same thing to your eye. You can see here that the eye makes this nystagmus. If you go round and round and round, and it makes a saccade, and then a slow movement, and then a saccade and a slow movement. The slow movement is coming from your VOR. So if you're turning faster, the VOR would turn your eye faster in the opposite direction. And also you see that sometimes the frequency of these saccades are occurring more quickly, more frequently. And that occurs when you try to look in the direction that you're turning. So this over here on top is the direction that you're turning. And if you want to look in that direction, you make these saccades more frequently. So that keeps your eye, if you're turning to the right, it keeps your eye in the rightward position. Okay. Sometimes you get the very same type of movement, but you've got a lesion. Okay. You can see here, again, you're getting what's called nystagmus. But in this case, there's a fundamental difference between what we saw before and what we're seeing now. In this case, we're getting nystagmus, but the head isn't turning. Can anyone guess where the lesion is? So the eyes are turning horizontally, so it's a horizontal canal problem. Is it the canal on the right or the left that has been damaged? Can anyone guess? Anyone come up with a solution? Yeah? Which side is it? The right side? If you're, okay, so we're turning to the right, okay? When we're turning to the right, which canal is more active? The one on the right, okay? Here, you got exactly the same nystagmus. Which canal is more active? The one on the right. So, but the head isn't turning. So how do you get the one on the right to be more active? Well, you lesion the one on the left. Okay? Yeah, so that's what's causing the nystagmus. This side here is producing less activity than this, and that simulates. Occasionally you get this strange form of nystagmus. You notice two differences here. You notice that instead of going smoothly back to between one saccade and another, this thing is going slowly back to this position. And it's changing direction. Sometimes it's going to the left, and sometimes it's going to the right. In this case, it's lesion of the PPH, your integrator, your calculus computer. And you make a saccade, and it sweeps back. Make a saccade again, sweeps back. Because you can't hold that lateral position. And then same thing happens if you move in the other direction. It sweeps back. Okay. Next, let's look at vergence eye movements. All kinds of things are happening here. You see here the eye moving from one position to another position as the target moves from near to far and then back again. This is what each eye sees, okay? And you can see all kinds of things happening to this. One thing, when the eye is like this, then both eyes are looking at the same target. Whenever the target isn't here, they aren't on the same target. The target is, you have disparity. The thing that makes you see either two objects or things in stereo. And the reason these things are serious because the eye hasn't moved very quickly. Okay. So it takes a little bit of time for the disparity to disappear. But you get these vergence movements. They're not very fast, as we saw, but they're fast enough, and the disparity disappears. The other thing you see is some blurring, okay? And because of this blurring, the blurring is because the lens has, you can see the lens here, and it also opens up, becomes rounder or flatter, or rounder or flatter, not instantly. It takes some time for it to change its shape. And so during that time, you've got blurring occurring. The third thing that happens is this pupil starts changing shape. And it does so sort of at the beginning of each movement, it seems. And it's trying to respond to the blurring, okay? Because when the target moves to a near position or a far position, it's blurred initially because it's at the wrong position for the eye or the lens. So the eye responds by making the pupil smaller, and so you see a crisper image, at least for a little bit of time. So it increases your depth of field. Okay, so vergence movement, two things can initiate them: a blurring or disparity, the difference between the two eyes. And if either of those things happen, it generates a vergence movement. Both those things can also change what your lens shape. So either a blur or a disparity. And the best thing, of course, happens when both are activating these two systems. Okay, the next movement we saw was pursuit. We saw early in the course, in the motion section of the course, that motion signals come from the eye, go to V1, go to our structure called MT, and then there's two parts of MST. In this case, we're going to MSTL, which is involved in tracking small objects. Okay? And that thing generates the pursuit eye movement. And that goes through the structure called the cerebellum for some reason, and then to the brainstem. And you can see here what the eye will see if you generate a pursuit movement. So first of all, the bee moves. Okay, you get a disparity. The eye starts following it. Then it makes a saccade to recenter the bee on the fovea, and then you have the eye the bee perfectly stable on the fovea for this part of the movement. We'll redo it again. So bee starts moving, eye starts tracking, saccade, tracking again. Okay, so it has these steps. This is the response it takes before the pursuit movement gets initiated. So it takes time before from when the eye sees it to when the signal leaves the brainstem. Okay, so this is the time it takes to travel through that circuit. And then it starts pursuing, and then you make a saccade to recenter it. Now, you can, in some circumstances, make a pursuit eye movement without seeing a moving target. Now, if you were to imagine, you can easily track these dots themselves. That's no trouble because they are a moving target. But you could, with a little bit of practice, imagine that these dots represented the center of this wheel here, and track it instead. So there's no not there in the center of its wheel, but with practice you can train yourself to do it. Also, if you were to close your eyes and underneath your eyes, you could monitor them. If you had something that moved back and forth, like a bee buzzing in front of you, back and forth, you could learn to track it with your eyes closed. And finally, you could do the same thing if you moved your finger back and forth in front of your eyes. You can learn to track them. Okay? Now this is amazing. This is, if you're looking at the back of the eye with really high magnification. And this would be the letter A projected on the back of the eye. Okay? And it'd be like a normal font that you'd be reading with. Notice those little circles there are your cones. Notice that this A isn't, the image of this A isn't stable on these cones. It's moving around across many cones, and it's moving around in two ways. First of all, there's these quick jumps. They're little tiny little saccades, and then a slow drift. Okay. Now you would think that these type of movements would cause the image to be blurred, but for reasons we don't understand, in fact, it helps you see them. Experiments were done where you could measure these tiny little movements and shift this A around just in the same distance as the eye moved. So you could then artificially stabilize the A on the back of the retina. And when this was done, the A disappeared. You became blind to the stable image. Okay? So for reasons that we don't know yet, these little movements of the eye that occur during fixation, you have no way of suppressing them, help you see. Okay, so we covered today that the function that you've got these movements, and they have different functions and different characteristics. And two of them, the VOR and the optokinetic system, they belong to an old system. They're movements that are present in the earliest species, like fish. And these newer systems evolved with the production of a fovea. So you have at the center of the eye that sees clearly. Okay, so you have these new systems, the saccades, the pursuit of divergence, to keep the eye stable on the fovea. In this case, it's keeping the eye stable on the whole retina. So the VOR does this. It's fast because its reflex is short, but it responds poorly to a continuous rotation. And the VOR is calibrated by the cerebellum. We have then the optokinetic reflex, and it used to help the VOR during the prolonged rotations. And this reflex is slow in getting going, but that's okay because your VOR is fast. And it's best elicited by everything moving on your retina, not just a small little target. If a small little target moves on your retina, you generate pursuit. Now saccades, they're part of the newer system. They point the foveas to an object of interest. They're fast and accurate, and they minimize the time that it takes for the movement, and thus maximizing the time in which the eye is stable. And you can't control its speed. Then you have pursuit, which lock in on a moving target. They're reflexive. Most cases you need something moving to elicit them. And they're slower than VOR because they have a longer pathway that goes through that MSTL loop. And then Verge's movement, they point each eye's fovea at the target if the distance changes. And without vergence, you see double vision diplopia. And the eyes move in opposite directions. Okay, that's it for today. Thank you very much.